Two cells. That's all it ever took. Your mother's egg, your father's sperm. They merged, divided, and kept dividing until somehow they became you. You've probably never questioned that process. It seems fixed. Biological law, the only way it has ever worked. But the question sitting quietly underneath all of modern biology is this: does it have to stay that way? Can humans, one day, make humans? Not in the way your parents did, not even in the way a fertility clinic does, but from scratch, from a cell, from nothing that resembles a beginning. The answer, right now, is no. But the distance between no and not yet has been closing faster than almost anyone expected. To understand where this is going, you need to understand where it started. The year was 1978. In a clinic in England, two scientists named Patrick Steptoe and Robert Edwards took a woman's egg, fertilized it outside her body, waited a few days, and placed it back in her womb. 9 months later, Louise Brown was born, the world's first test tube baby. The reaction was not calm. Newspaper headlines called it unnatural. Religious leaders said science had crossed a line. Ethicists warned that the definition of human life itself was being dismantled. But Louise Brown is 47 years old today. She has children of her own. And more than 12 million people alive right now were born through IVF. Robert Edwards received the Nobel Prize in 2010. What a generation called unthinkable. The next generation called Tuesday. But IVF was still just a shortcut. Sperm still met egg. Biology still ran its usual course, just in a different location. The next step was stranger. In 2006, a Japanese scientist named Shinya Yamanaka took an ordinary skin cell from an adult mouse and reprogrammed it into something that behaved like an embryonic stem cell. Not a new cell, not a lab-engineered cell, a skin cell. The kind your body sheds by the thousands every single day. And he reversed it, wound it back to the beginning. That work won the Nobel Prize in 2012. And it suggested something that nobody had seriously entertained before, that every cell in your body still carries the instructions for becoming anything. The information isn't gone, it's just switched off, which raises an uncomfortable question. If any cell can be rewound to the start, what exactly is special about the egg and the sperm? In 2023, researchers at the Weizmann Institute, led by Jacob Hanna, guided human stem cells into forming a structure that closely mirrored a natural embryo in its first 2 weeks of development. No egg, no sperm, no fertilization. The cells just organized themselves, the way cells do, when given the right chemical signals and enough time. It wasn't a human. It couldn't develop further, but it looked like a beginning. And here is where the speculation becomes reasonable rather than science fiction. If a cell can be reprogrammed to its embryonic state, and if stem cells can self-organize into embryo-like structures, then the logical question is, what's actually stopping the rest? Not a biological wall, a series of technical problems. And technical problems historically get solved. The barriers right now are real. We don't know how to sustain development past the earliest stages outside a womb. We don't know how to replicate the full biochemical environment a developing embryo needs. We don't know how to build a synthetic placenta that actually works. But none of those are mysteries of nature. They're engineering problems. Think about what that framing means. 100 years ago, powered flight was also a collection of engineering problems. 50 years ago, so was putting a person on the moon. 20 years ago, sequencing a human genome took a decade and cost $3 billion. Now it takes a day and costs 1,000. The trajectory of biology has been the same as every other technology. What seems impossible becomes difficult. What seems difficult becomes routine. What seems routine becomes cheap. So if the question is whether humans can ever truly make a human, the honest answer is probably yes. Not soon. Not without decades of science and decades of ethical argument about whether we should. But the biology is not saying no. It's saying, not yet. And not yet has a very different meaning than never. Here is what makes that strange to sit with. There is a cell in your arm right now, doing nothing, dividing occasionally, dying eventually, being replaced. And inside that cell is the complete instruction set for a human being, your genome. Every decision your body made about you, written in the same language as every other human who has ever lived. With enough knowledge, someone could theoretically take that cell, strip it back to its earliest state, coax it into organizing, sustain it, and grow something. Something that carries your DNA, something that starts the way you started. Not you, never you, but something that begins the same way you did. That is not a horror story. It's a question. One that biology is slowly, methodically forcing us to answer. Because the definition of what makes a human has always shifted. We drew the line at natural conception, then IVF moved it. We drew the line at 2 genetic parents, then cloning research moved it. We drew the line at biological starting materials, and now synthetic embryo models are sitting right at the edge of it. Every time we said this is what a human is, biology eventually had more to say. Those 2 cells that started you didn't make you. They just switched something on. But what gets switched on, it turns out, might not need them forever.